Okay, 3, 2, 1, it's jam.
What's up, science fam? Making your own food can be fun, but it's not always simple. It's not always easy. That's why the fast food business is booming, especially during these times. But what about plants? They're cheating. What exactly allows plants to make food and oxygen for themselves? My name is Saramon, and this is Science with Saramon, and our topic is photosynthesis. So let's get right into it. Photosynthesis is how plants and other animals and some forms of bacteria make food on their own. So how exactly do they do that? So we're going to discuss in this session the importance of chlorophyll and other pigments, describe the patterns of electron flow in the light reaction events, describe the Calvin cycle, and differentiate different photosynthetic pathways. In essence, we're going to talk all about photosynthesis. So consider this tree. Behold, it's a tree. You look outside your window, you're probably going to see trees or other forms of plants, and most of what you're going to see, most of the surface area that you see in a properly growing tree would be the thing that gives shade. So what is it that makes good shade in a tree? That would be the plant organ, which is leaves. Leaves are wide and they're flat most of the time, and that's because their structure relates to their function, which is to catch sunlight, specifically for photosynthesis. Now, what exactly is photosynthesis? We'll get to that. First, what makes leaves green? The leaves are green because of an organelle found in their cells called the chloroplasts, and these are the specialized organelles for photosynthesis. And they have many parts, you can see them here, but of note are the thylakoids. These are flattened sacs that are basically folded inner membrane sacs. So they're folded together into tiny sacs, all right? And this stack of folded sacs are would be called a granum or a grana. So a granum is the singular, grana is the plural. The stroma is the soup inside the chloroplast, and it has an inner and outer membrane. That's why the inner membrane can fold into thylakoids. Now the thylakoids are covered in enzymes, and that's why you want to fold them into these flattened sacs so that you have more surface area and more area for the enzymes to be on. And again, enzymes catalyze reactions, and that allows us to have lots of enzymes doing their jobs, doing their work. So what makes the chloroplast green? What pigment is in the chloroplast? It's chlorophyll. Chlorophyll is a pigment found on the thylakoid membrane, so it's on the thylakoid membrane itself, and it captures light. So chlorophyll isn't the only pigment, which we'll see later on. So what is the purpose, the main purpose of photosynthesis? It's basically to address the needs of plants, and this is the general chemical equation for photosynthesis. So it takes carbon dioxide, it takes water, and it takes sunlight in order to produce glucose and oxygen. So this is the chemical formula of glucose, and this is of course oxygen gas. So, of course, since you guys probably have chemistry already, you have to balance the chemical equation for photosynthesis. And to balance it, all you have to do is put the number 6 before all of the small ones, and that balances out both sides of the chemical equation. We have 6 carbon dioxide molecules, 6 water molecules, and 6 oxygen molecules in order to get all of that relates to 1 glucose molecule. So what kind of reaction is this? We've talked about multiple kinds of reactions. Some require energy, some release energy. Some of them build up materials, some of them break down materials. This is basically an endergonic anabolic reaction, meaning it is a metabolic pathway that builds up, it synthesizes complex molecules, specifically glucose, and it requires energy. And the required energy is supplied by sunlight. That's why it's photosynthesis. It's light being used to create complex molecules. So light itself is both waves and particles. It has a dual nature of light, and these particles are called photons. Now, the reason why plants try to capture light as a source of energy is because of this dual nature of light, and because it's a form of electromagnetic radiation. They're waves made of electrical and magnetic energy. So there's lots of energy in light itself. And when we talk about light, different wavelengths of light or different distances between the waves determines the color of light, and that's related to your pigments. So lower wavelength usually means higher energy. That's why the lowest wavelengths, which is near violet, are the highest energy. That's why ultraviolet light itself is very high energy to the point where it can become damaging to certain parts of your cells. Whereas red light is very low energy because of its very long wavelength. So different colors of light bend at different angles, and you can see here that the red light bends the least. And that is related to why red light is something that is important for plants. So different things have different colors because whatever they absorb is absorbed, whatever they reflect is what we see as their color. So again, we only see colors reflected by objects. So that relates to the color of plants. They're mostly green, and that's because they reflect green wavelengths of light. So this is an absorption spectrum diagram, meaning it shows you how much chlorophyll absorbs a certain amount, a certain wavelength of light. So you can see it absorbs mostly in the wavelengths that are near blue and near red. And the reason for that is because, again, short wavelength or near blue light has high energy, while red light, the red color of light, the long wavelengths of light, will travel the farthest throughout the Earth's atmosphere. That's why at sunset there's usually red, and the color of the sky is usually near red during sunset because, again, red light travels the farthest, and so it lasts the longest throughout the day. So your chlorophyll is specialized to get the high energy light as well as the light that lasts until evening, your blue and your red light. So these are other examples of pigments. It's not just chlorophyll. We also have a pigment called anthocyanin, which reflects red, purple, and blue light. So if you've seen purple leaves, which are not that uncommon, especially with landscaping nowadays, that has anthocyanin. And of course, your carotene in carrots, it reflects red and orange light. So that allows it to absorb different wavelengths of light, which can allow certain species of plants to adjust to different areas or different conditions. So what is the main purpose of pigment molecules? Again, it's to capture light. So they capture light energy for photosynthesis, and that fuels the light-dependent reactions because, again, light-dependent. They depend on light. So it harvests energy from light and electrons from water in order to produce something. So this entire diagram, we're going to go through this step by step. All right, so let's do this. Let's start first with these two terms. There's P680 and P700. They are also referred to as chlorophyll A molecules. These are special chlorophyll molecules, which we'll see in the diagram when we go close up to it. There's also two special enzymes, ATP synthase and NADP plus reductase. So the names of enzymes are very much related to their function. So ATP synthase, from its name, it synthesizes ATP, while NADP plus reductase, it reduces NADP plus. So we'll see more of that when we go through the diagram. So let's focus on this part of the diagram for now, this first part, the one labeled PS2 or photosystem 2. Here we see the first chlorophyll A molecule, your P680. It has an important function there. So let's discuss this. So in photosystem 2, it's a special group of proteins that catch and process light. So again, a photosystem is a group of proteins that catch and process light. So first, light in the form of sunlight, your photons, will excite the electron that's on a chlorophyll molecule. So again, the pigment molecule, its electron gets excited, and because this electron is excited and has high energy, it gets sent away. And what does the sending is your chlorophyll A molecule, your P680 molecule. It sends that electron, that high energy electron, to an electron acceptor, which is somewhere over here. We're not going to discuss the specific name. Just remember, it's just caught in like an oven mitt. Okay? It's like a hot potato caught by an oven mitt, and it's waiting to go to the next stage. But again, if an electron is sent away from a chlorophyll molecule, that means there's a missing electron on your chlorophyll, and we need to replenish or replace that missing electron. So what we do is we break down water in order to replace that electron through a reaction called photolysis. So photolysis is the breakdown of water using light energy. That's why it's photo, light, and then lysis is breakdown. So we break down water to make oxygen gas, 4 hydrogen ions, and 4 electrons. So, oxygen is literally just a waste product of photolysis. And the electrons are what the photosystem needs to replace the missing electrons. Now, these hydrogen ions will be important in the next stage. So again, just to emphasize, oxygen gas is a waste product of photolysis. Your plant does not use it in photosynthesis, right? Not in photosynthesis itself. So we're done with PS2 or photosystem 2. Now we go to the middle part. So this middle part here is called the electron transport chain of photosynthesis. These are a set of enzymes, a set of proteins between photosystem 2 and photosystem 1. So the fourth step, so again, remember our excited electron, which is in the oven mitt here, it gets passed to the electron transport chain. So this excited electron will be going into the electron transport chain, and the energy of this excited electron will be used to pump hydrogen ions into your chloroplast's inner membrane. So again, this downwards thing, it goes from the stroma going into the thylakoid. So it's pumping hydrogen ions into your chloroplast's inner membrane. And step number 6, your hydrogen ion movement will give ATP synthase the energy to make ATP. So again, when your hydrogen ions get pumped into your chloroplast's inner membrane, it creates a high concentration here inside the inner membrane, and it will eventually diffuse back outside. And when it diffuses back outside, this is coupled with your ATP synthase. So the diffusion of hydrogen ions back out will power your ATP synthase. So your ATP is your adenosine triphosphate. It's the energy of your cell. So that's the first product that we're gonna take note of, ATP. So again, ATP is one of the products of your light-dependent reactions, and it's made in the electron transport chain. Now let's move on. Your electron transport chain has these coupled processes. So when we say coupled processes, that means that a passive thing is powering an active thing or something that requires energy. So the exergonic fall, this is just a complicated term for your electron, your excited electron giving off its energy. So your excited electron, when it gives off its energy, that's used to pump your hydrogen into your chloroplast. Now, your hydrogen chemiosmosis is the complicated term for the diffusion of hydrogen ions back out. Again, back out of your chloroplast. And that passive transport will power your ATP synthesis. So again, two pairs of coupled processes will end up creating ATP for your plant or for your photosynthetic organism. So now we move on from the electron transport chain to the final stretch of your light-dependent reactions, your PS1 or your photosystem 1. Right? So your photosystem 1, so why is it labeled 1 while the one that came before is 2? That's because this one was discovered before photosystem 2. It's just by chronological order of discovery, not by their function. But anyway, photosystem 1 is very much like photosystem 2, but it can recycle. So the light excites the chlorophyll electrons here as well, but in this case it has P700, another special chlorophyll A molecule. So this P700 sends that excited electron to an electron transporter where it's held again, and that held electron has two possible ways where it can go. It can go either forward or backwards. We'll discuss that in a bit. But first, that missing electron in P700 has to be replenished, and that's replenished by the electron, it can be replenished by water being broken down or the electron that's coming from the electron transport chain. So once again, your electron transport chain, the depleted electron can be used to replace the missing electron. So it recycles that electron. So there's two ways that the accepted electron can go. It can go forward or backward. When it goes forward, it is taken up by NADP plus reductase and put into NADP plus to become NADPH. Now, NADPH is basically an electron transporter. Just think of it like a tricycle or a taxi. it picks up its passengers, which are the electrons, the excited electrons, and it shuts the door of the taxi using a hydrogen ion to become NADPH. So that's the forward way of where the excited electron can go. The backward way is that this excited electron can actually go back into the electron transport chain. So again, it can go forward and out through the electron transporter, or it can go back to the electron transport chain. Now, what's the purpose of this? It produces two types of products. There's the ATP, which is from the electron transport chain, and the NADPH, which carries electrons. So again, it can produce energy and electrons. If your cell is lacking in ATP or energy, it can go back to the electron transport chain, triggering what we call a cyclic flow. So it's just a cycle where the electron keeps flowing through the electron transport chain. Or if your cell is more lacking in electrons, it can go forward into the non-cyclic flow, or basically being carried off by NADPH. So these two things, energy and electrons, these are necessary reactants for your next phase of photosynthesis. So let's discuss more about NADPH. This is the chemical equation of your electron transporter. Your sorry, there's a typo there, that's supposed to have a P. Your NADP plus will combine with two electrons, and it will pay the fare or shut the door with a single hydrogen ion, and that balances out the charges. That's why it became NADPH. So if we have light-dependent reactions, we should, of course, have light-independent reactions. So these are reactions that are part of photosynthesis and don't need light. So just because it's light-independent doesn't mean it has to be in the dark. It just means that it doesn't care whether light's there or not. So these reactions come in a cycle, which is called the Calvin cycle. It occurs in the stroma. So it's occurring in the liquid part of your chloroplast. So the Calvin cycle has three stages: carbon fixation, reduction, and regeneration. And we're going to discuss these stages one by one. So first we have carbon fixation. Literally, it's to fix carbon from carbon dioxide into a compound. And what does this fixing is Rubisco, an enzyme that makes carbon fixation possible. So Rubisco is one of the most important enzymes to life in general, because without it, there would be no such thing as photosynthesis. So what happens in carbon fixation is that your RuBP, or ribulose biphosphate, is being fixed with carbon from carbon dioxide to become an unstable 6-carbon compound. So this doesn't last very long, so we don't need to know its name. But basically, your ribulose biphosphate, a 5-carbon compound, you can see these 5 circles here, 1, 2, 3, 4, 5, gets fixed with another carbon from carbon dioxide. So 5 plus 1 equals 6. And this 6-carbon compound is unstable, so it breaks down into 3-carbon compounds instead. So those 6-carbon compounds become a 3-carbon compound called 3-PGA, or 3-phosphoglyceric acid. So it's called 3-PGA because there's a phosphate group on the 3rd carbon. So you can see these diagrams here. There's 3 carbons. If you number them 1, 2, and 3, the phosphate is attached to the 3rd carbon. So it's 3-PGA. So Rubisco is called ribulose 1,5-biphosphate carboxylase oxygenase. And again, enzymes have their names usually pertaining to their function. So again, your ribulose biphosphate is being carboxylated, or having carbon affixed to it from carbon dioxide. So it catalyzes again ribulose biphosphate, a 5-carbon compound, with carbon dioxide to form an unstable 6-carbon compound, which breaks down into 2,3-PGA molecules, right? So again, our product after carbon fixation is 3-PGA molecules. Next, we have the reduction stage. So in reduction, the meaning of reduction is the gain of electrons. So, sir, it's reducing, so why would it be gaining? So if you haven't taken it up in chemistry, one way to remember it is that reduction is like it's reducing the charge. All right, so it's adding more negatively charged electrons to your molecule. So this is where your NADPH, your electrons from the light-dependent reactions, and your ATP come into play. So your ATP and your NADPH, your energy and your electrons are shoved into 3-PGA to turn them into G3P, or glyceraldehyde 3-phosphate. So it looks very similar to your 3-PGA, only because this is a simplified diagram. But again, the 3P means that the phosphate is attached to the third carbon molecule. So G3P is glyceraldehyde 3-phosphate, and it's used to make glucose. So if, again, to summarize the reaction 3-PGA, when it's phosphorylated, when you add ATP, it becomes what we call 1,3-BPGA, or 1,3-biphosphoglyceric acid. So from 3-phosphoglyceric acid, it became 1,3-biphosphoglyceric acid. It just means there's an extra phosphate group, and it's placed at the carbon 1 and carbon 3 positions. So again, the name relates to what it looks like. Now your 1,3-BPGA, when it's reduced, it becomes G3P. Okay, so that's pretty much what's happening in the reduction phase. So again, your two G3Ps, if you put them together, you can make a glucose. So your G3P with the 3 carbons, you get 2 of them together, you have 6 carbons in total. That's your C6H12O6, or your glucose. But only one G3P for every turn of the Calvin cycle is used to make glucose. All the rest, all these 5 leftover ones, are used in the third stage of your Calvin cycle, your regeneration stage. So in regeneration, this is just to reset the cycle. We need your RuBP back. So your RuBP, your 5-carbon molecule called ribulose 1,5-biphosphate, it is regenerated from 5 G3P molecules. And you can see the logic of it if you count the number of carbon atoms. So there's 3 carbon atoms here in G3P. If you have 5 of them, 5 times 3 is 15. So we have 15 carbons in total. If we rearrange those using energy from ATP, you should get 15 carbon atoms that are distributed to 3 molecules. So 15 divided by 3 would have 5 carbons in each molecule. So all of these carbons were just rearranged to form RuBP, or ribulose 1,5-biphosphate. Right? So that's your regeneration stage. So again, 5G3P is used to regenerate 3 RuBP. 5 times 3 is 15. That's 15 carbon atoms. And 3 times 5, meaning 3 RuBP molecules with 5 carbons each, is 15 carbon atoms. All the accounting is there. So again, Rubisco, its full name is ribulose 1,5-biphosphate carboxylase oxygenase. So it's not just fixing carbon. It's doing this sneaky thing, oxygenase. So it's not just carboxylating, it's also oxygenating. What is that? That is basically photorespiration. It's the opposite of photosynthesis when Rubisco binds with oxygen instead of carbon dioxide. So there's no carbon to add there. So instead, it does something like cellular respiration, and it's a big waste of time and energy. So your plant wants to avoid this. It wants to avoid Rubisco doing this waste of time. So some plants have adaptations to reduce photorespiration. So if you count the number of carbons in the first stable compound of the Calvin cycle, there's 3 carbons in each one. So the first photosynthetic pathway, or the first way of doing photosynthesis, is named after this first molecule, your 3-PGA. So it's named after that. So it's called the C3 pathway because there's 3 carbons in the first stable carbon compound. So it's the regular pathway. It's 0 modifications. It just looks like this. So pretty much your Calvin cycle is happening in the same cell as everything else. Rubisco has a chance of doing photorespiration. So again, this is not ideal if your cell is in a climate where Rubisco would bind with oxygen instead of carbon dioxide. So this is especially true in high temperature environments, specifically tropical places like here, the Philippines. So your C4 pathway is a different pathway. It minimizes photorespiration by separating processes into different cells. So this is how it does it. So first, carbon fixation happens in a different cell using a different enzyme. But it's not going to produce 3-PGA. That's why it still needs Rubisco later on. So what happens is PEP carboxylase, or phosphoenol pyruvate carboxylase. Again, the enzyme is named after its function. So what it does is that it's an enzyme that never binds with oxygen, but it can only fix carbon dioxide to phosphoenol pyruvate, a 3-carbon compound here that is not very ideal for making 3-PGA. So instead, it becomes oxaloacetate, and from oxaloacetate, it'll become malate. Now, this malate is the stockpile of carbon in your plants. So this is stockpiled in your mesophyll cells. So again, in your mesophyll cell, your PEP carboxylase just stockpiles carbon in the form of malate. Now, later on, your malate is transferred to what we call a bundle sheath cell. And this is where the rest of the Calvin cycle takes place and where Rubisco does its job. Now, the bundle sheath cell is special because it doesn't have any contact with the atmosphere. It doesn't have any contact with oxygen gas in the first place. So Rubisco has no choice but to work with carbon dioxide that was stockpiled from malate. So again, in your C4 pathway, there's extra steps. It uses a different enzyme and it has to transport malate to an entirely different cell. Okay? So it costs extra ATP because of the transporting and the extra enzyme, but it's worth it in warm places. Why? Because Rubisco, again, has no choice but to bind with carbon dioxide. Next, if we go to the deserts, your cacti and other desert plants face the challenge of water. Water is in extreme scarcity in your desert, so they need to conserve water. So they have a special photosynthetic pathway where it varies by day and night. So you lose more water, usually, of course, during the day in the desert because it's hotter. So for your desert plants, they have the CAM pathway, or the crassulacean acid metabolism pathway. Complicated term, but basically the concept is that their pores, their stomata, are closed during the day so that they don't release water, but they're open during the night. And it also avoids photorespiration by employing some of the same strategies as the C4 pathway, but this time it's just separated by time instead of separated by different cells. So at night, your carbon fixation happens while the stomata are open. So your carbon fixation happens using PEP carboxylase. Same thing. It takes PEP, your phosphoenol pyruvate, and then it fixes carbon from carbon dioxide to make oxaloacetate, which turns into malate or other organic acids. Now, this malate or other organic acids will go to the Calvin cycle during the day. So again, at day, the Calvin cycle, the light independent reactions, again, even though it's day, it's light independent. It doesn't need light, so but even if it's there, it can still do it. Your Calvin cycle works while the stomata are closed. So again, since the stomata are closed, there's no oxygen gas that is exposed to Rubisco inside the cell. And the carbon from carbon dioxide usually will be taken from malate instead. So again, same concept. Stockpile your carbon and malate and then use it up in the Calvin cycle later on. But in this case, it's not different cells, it's just different times. So like C4, this keeps oxygen from Rubisco. Very efficient. Keeps the water in and keeps the oxygen out. So photosynthesis, it's complicated, but we study it because it's the engine of life. All right, so that's because solar energy, the main source of energy for all biodiversity on the world, can only be harnessed through photosynthesis. So the fuel of the sun, the solar energy of the sun, what bridges it to the rest of life is photosynthesis. So that's pretty much it for this topic, and I'll see you guys next time.
Party's over.